Today, we are going to talk about phytohormones in defense against abiotic stress. Plant hormones or phytohormones are biochemicals required for the normal growth and development of plants. Plant hormones include auxins, gibberellins, cytokinins, abscisic acid, ethylene, besides jasmonic acid, salicylic acid, brassinosteroids, strigolactone, and nitric oxide. Apart from their role in plant growth and development, hormones also mediate response to biotic and abiotic stress. Biotic stress, like diseases, pathogens, herbivores, etc. Abiotic stress, like drought, heat, salinity, heavy metals, right? Hormones act at the site of their biosynthesis or some distance away from it. Hormone biosynthesis, distribution, and patterns of their signal transduction change under stress conditions. Ethylene and abscisic acid play remarkable roles in the regulating the abiotic stress response. Abiotic stress. Ecological factors favor plant growth at optimum levels and constitute stress at sub or supraoptimal levels. Abiotic stress reduces crop productivity by about 50%. High temperatures lead to 20% decrease in the yield. Low temperatures 7%, salinity 10%, drought 9%, and other forms of stress cause 4% yield loss. In grain crops, grain size, number, and dry weight are influenced by abiotic stress, especially if present during the reproductive phase. Crop productivity may be reduced by 2.5 to 16% by 1 degree Celsius rise in seasonal temperature in tropical and subtropical regions. The stress response depends on the genetic constitution and the adaptive response of the plant. First, we'll start with the drought, that is hormones in defense against drought. Drought has been defined as a period of abnormally dry weather, sufficiently prolonged for the lack of water to cause a serious hydrologic imbalance in the affected area. It is one of the dominant factors diminishing crop productivity. All aspects of plant growth, including photosynthesis, protein synthesis, water relation, cell trajectory, then membrane integrity, and nutrient uptake, are affected by drought. It also causes oxidative stress and damages the biological molecules, including DNA, proteins, and photosynthetic pigments. In response, plants synthesize a range of molecules as protection against drought stress. For example, proline, glycine, betaine, soluble sugar like mannitol, sorbitol, trehalose, then polyamines and proteins. ABA levels, abscisic acid levels, means uh, regarding hormones. Abscisic acid levels increase in plants under drought stress, including the expression of ABA-dependent genes. ABA signaling leads to the closure of stomata, reducing transpiration. Expression level of some genes like ZDP, zeaxanthin epoxidase gene, AAO3, Arabidopsis aldehyde oxidase gene, NCED3, 9-cis epoxy carotenoid dioxygenase gene, and the MCSU, molybdenum cofactor sulfurase genes are increased upon osmotic stress. ABA, abscisic acid, is transported into the guard cell through passive diffusion via members of ABC and nitrate transporter families. ABC family members like including ABC G25 and ABC G40 and nitrate transporter means genes belonging to nitrate transporter families A1, IT1, NRT 1.2, and NPF 4.6. Right? ABCG25 is an ABA exporter with tissue-specific expression induced by ABA and drought stress. ABCG 40, AIT1, NRT 1.2 and NPF 4.6 import ABA into the guard cells. ABA also generates reactive oxygen species ROS, which leads to increased cytosolic calcium ion levels and stomatal closure. About 14 ABA receptor proteins mediate ABA signaling. Pyrbactin resistance 1, PYR1 and PYR1 like regulatory elements undergo a conformational change after ABA binding and inactivate the clad A serine threonine protein phosphatase 2C, PP2C. This in turn triggers the ABA signaling cascade by phosphorylation of serine threonine kinases. Now, I will tell you some results or some findings of various studies. It has been observed that salicylic acid biosynthesis is also suppressed at high temperature in tobacco after TMV infection and in Arabidopsis after PST 350 DC 3000 infection. High temperatures suppress the expression of isochorismate synthase 1 enzyme. Isochorismate synthase 1 do not show temperature sensitivity to infection. Then jasmonic acid biosynthesis genes are upregulated by moderately high temperature, moderately high 29 to 30 degrees Celsius after wounding in Arabidopsis, means it has been observed in Arabidopsis, right? Next is temperature. Temperature affects the distribution, phenology, and physiology of plants. Temperature is increasing under dry as well as wet conditions in the changing global climate scenario. The crop productivity decreases by 6% for 1 degree rise, as I already told you. 1 degree rise in temperature beyond the optimum. Temperature stress causes accumulation of reactive oxygen species, then denaturation, misfolding, and aggregation of proteins, changes in membrane structure affecting permeability and raft distribution. Besides, it impacts on leaf area, leaf retention, stomatal conductance, water potential, and rate of transpiration, right? Photosynthetic capacity may be damaged or permanently damaged due to heat stress. Plants produce transcription factors, heat signaling, proteins, and molecular chaperones to prevent protein misfolding and aggregation after heat shock. In response to heat stress, the endogenous ABA levels increase, transiently increasing the antioxidant capacity. For example, by inducing RBOH NADPH oxidases. Out of 10 different RBOH genes identified in Arabidopsis, only RBOHD is upregulated in response to heat stress. It has been observed in some studies. The mutants for this gene show low germination and seedling survival at high temperatures. Now, what are mutants? A mutant is a plant that has a different gene structure than other plants of the same type, right? So, ABA biosynthesis inhibitors and ABA signaling mutants have impaired heat stress tolerance. Both the heat shock proteins and their transcription factors are regulated by abscisic acid. So, expression level of ABA1, ZDP, and NCED259 increase in Arabidopsis at 32 degrees Celsius, increasing the ABA levels. ABA may also modulate carbohydrate and energy status to strengthen the heat stress response. Ethylene is another hormone involved in heat stress tolerance. Studies have indicated increased synthesis of ethylene under heat stress. EIN2 and ER1 mutants have poor survival rates under heat stress. However, it has been observed that in Arabidopsis, EIN2 to 1 mutants exhibit greater tolerance to heat stress. Ethylene is also involved in carbon dioxide-induced heat stress responses in tomato plants. That is, this has been observed in plants, tomatoes, and increased thermotolerance in rice. Cytokinins also play an important role in heat stress response in plants. They increase the activities of APX, ascorbate peroxidase, SOD, that is superoxide dismutase, and GP, and also upregulate genes responsible for photosynthesis and carbohydrate metabolism under heat stress. GA biosynthesis, gibberellins biosynthesis, and accumulation also increase under elevated temperature in some plants. Next is salinity. Salinity has affected more than 800 million hectares of land globally, decreasing potential agricultural land by 1 to 2% per year. What is salinity actually? Soils with electric conductivity higher than 4 TS per m at 25 degrees Celsius and these are classified as saline, right? So more than 50% of the land in developing countries, particularly that falling in the arid region, is affected by salinity, causing yield losses to the tune of 40%. For example, in the case of wheat, it decreases the quantity as well as quality of the produce. Salinity impairs water uptake and causes ion toxicity, osmotic stress, nutrient deficiency, and oxidative stress. Salt stress causes physiological drought, impairing protein and photosynthesis. Changes in the intracellular calcium ion levels, then excess sodium and ROS accumulation are the signals that trigger the salt stress response. In response, ethylene ethylene is the major hormone in the salt stress response. In response, the levels of ethylene, as well as its precursors, ACC, means 1-amino-cyclopropane 1-carboxylate, increase under salt stress. Ethylene signaling involves 5 ethylene receptors. 5 ethylene receptors, a protein kinase, and key positive regulators like EIN2, which signals primary transcription factors, right? And primary transcription factors, EIN3, EIL1 means ethylene insensitive like 1 and EIL2, and many downstream ethylene response factors, right? 5 ethylene receptors are ETR1 means ethylene response 1, ERS1 ethylene response sensor 1, ETR2, EIN4, right? Ethylene insensitive 4 and ERS2, right? So during short and long-term salt stress, ethylene receptor genes ETR1, ETR2 and EIN4, signaling genes CTR1, EIN3, ERF1 and ERF2, and MAPK cascade genes like MEKK1, MKK2, MPK4 and 6 are upregulated in some plants, right? Accumulation and transcriptional activity of EIN3 and EBF1, EBF2 degradation are promoted under salt stress. Thanks for watching. If you liked this video, make sure to subscribe for more. Thank you.